Hey, shovel face, you want mine, Russell?
In this paper, we have shown that glyphosate, as an amino acid analog of glycine, may be erroneously misincorporated into polypeptide chains during protein synthesis. The research literature documents evidence of severe protein impairment through substitution of conserved glycines by other amino acids. It leads to the disruption of function of many proteins with essential roles in metabolism and regulatory processes.
Actually, no, I'm alright. I was thinking of chilling out by the throne for a little bit.
I'm gonna mind wrestle you whether you like it or not.
Well, I have to warn you, I'm getting pretty good at it.
Glyphosate substitution for conserved glycines and essential proteins can explain the destruction of glands and organs revealed by Monsanto, the original patent holder's own studies. Glyphosate is pervasive in the food supply, and chronic exposure will lead to slow accumulation of damaged proteins systematically. Fibrily plex and tangles intransigent to proteolysis may be due to glyphosate substitution for conserved glycines, accounting for multiple neurological diseases. Impairment in dimerization, membrane attachment, cytoskeleton attachment, and active site flexibility are some of the defects we anticipate. Some consequences are impaired fatty acid release, leading to obesity, impaired insulin receptor response, leading to diabetes, impaired 1-carbon metabolism, leading to neural tube defects and autism, impaired oxidative phosphorylation, causing mitochondrial disorders, impaired RF2 regulation. RF2, guys, is a basic leucine zipper protein that regulates the expression of antioxidant proteins that protect against antioxidant damage triggered by like injuries or inflammation.
Then I guess I can't make you do this.
Okay, mercy, you win. I give. Game over.
Leading to hyperkeratus and fatty liver disease, impaired cell cycle control during DNA synthesis, impaired collagen cross-linking, and dysregulated phosphorylation cascades leading to cancer, lung disorders, and autoimmune disease. These effects easily account for the multitude of diseases and conditions whose incidence is rising in the USA and elsewhere in step with the rise of the use of glyphosate on core crops. We urge regulatory agencies worldwide to take action to remove these synthetic amino acids not only from the food supply, but from our biosphere. I want to read a portion of the study here to you. DNA damage, chromated deletions, and achromatic lesions. Okay, one of Monsanto's early studies involved examining DNA damage in bone marrow of mice exposed to glyphosate. An interesting finding was a substantial increase in the number of chromated deletions and achromatic lesions observed following glyphosate exposure compared to controls. Achromatic lesions, gaps, and chromatides are induced by endonucleases that play a role in the repair process. The gaps are a manifestation of unrejoined DNA double-strand breaks following endonuclease activity. Citation 38. A possible explanation for the observed lesions involves impaired DNA repair mechanisms, particularly, concerning the nucleotide guanine. That's 7 and 8 dihydro 8 oxyguanine 8 oxo-G, for in other words, OXO-G, is one of the most commonly formed oxidative lesions in DNA. It's particularly destructive because it mispairs with adenine during replication, changing guanine and cytosine to thymine and adenine. Premutagenic lesions accumulate in mice that are defective in the gene coding the DNA glycolase enzyme, OGG1, which excises 8-oxoguanine from DNA. Citation 40.
What are you boys doing? Nothing, Dad. Just hitting the old punching bags.